folks. This is spring 2020. We're working on our unit for metabolism. We're on the second PowerPoint B. There's been some problems getting the PowerPoints posted to YouTube and then getting the links to work. So I thought I would try to do a really short, quick video on anaerobic respiration, and then let me see if I can get it posted to YouTube and then make it available to the public. We're just trying to troubleshoot here. So we will presume that we've covered aerobic respiration. So we're going to go to this very primitive form of respiration called anaerobic respiration. Now, most folks in biology haven't heard of this because it's a process that only some prokaryotes can carry out. Often there's some confusion in anatomy and physiology. Sometimes anaerobic respiration might be used for what we in microbiology would call fermentation, like lactic acid fermentation. So we just want to try to make sure that we clear up any confusion there. So with anaerobic respiration, let me see if I can back up here. Anaerobic respiration. So this is around slide 43. Okay, so let's see if we can't open this little guy up. All right, so in anaerobic respiration, folks, as microbiologists, whenever we see respiration, we know there's an electron transport chain involved. Anaerobic respiration as a microbiologist is telling me that we have an electron transport chain, but oxygen is not the final electron acceptor. We're going to use some alternate molecule. Now, this was the first type of respiration to evolve because you'll remember that initially Earth's atmosphere was anaerobic. So initially there wasn't any molecular oxygen around. It uses a different electron transport chain, different from the electron transport chain found in aerobic respiration. ATP yield is going to be less than in aerobic respiration. It'll be more than the 2 ATP per glucose we saw in fermentation, but it will be less than the theoretical max of 38 ATP we see in aerobic respiration. The two alternate electron acceptors I'd like you to know for anaerobic respiration will be nitrates, and some microbes, depending on their capability, can reduce nitrates to nitrites, and then some microbes can reduce nitrates all the way to molecular nitrogen. And such microbes would be called nitrate reducers. We'll see that E. coli is a fantastic nitrate reducer. Now, if we were farmers, and we know that our plants can use nitrate, and indeed sometimes as farmers we might add nitrate-containing fertilizers to our fields to help our plants grow, we wouldn't want this nitrate reduction to occur because it would be taking usable nitrate that our plants can use and reducing it back to the unusable molecular nitrogen. So this process that we'll see in the nitrogen cycle is called denitrification, is something that farmers wouldn't like. However, if we're, say, working in sewage treatment, we like nitrate reduction for treating sewage because we want to get rid of all that usable nitrogen in the sewage, the treated sewage, before we dump it into our waterways or coastal waters. And the reason we want to get rid of usable nitrates, say, in treated sewage is if a lot of treated sewage has nitrates, when we dump it into our waterways, algae and cyanobacteria can use it as a source of nitrogen to grow and multiply like crazy and make what are called these blooms. So when we have excess nitrates in our waters, we get what are called harmful algal blooms, which is an increase in the population size of algae and or cyanobacteria. And these harmful algal blooms are bad news for two reasons. Some of the algae and cyanobacteria will make toxins, which can be poisonous to aquatic organisms, can be toxic to humans. You probably heard of the notorious red tides. That's an example of a harmful algal bloom. Sometimes shellfish, which are filter feeders, will consume the toxin-producing algae, and the shellfish aren't hurt by the algae, but the toxins accumulate in the tissues of the shellfish. So if humans come along and eat them, or marine mammals come along and eat them. Even sometimes birds can be affected. They can get a toxic dose. So again, if we're working in sewage treatment, we might want to encourage nitrate reduction to get rid of those usable nitrate in the treated sewage. Furthermore, you guys, if we are farmers, sometimes if we're using excess nitrate-containing fertilizers, if it rains or we over-irrigate in the runoff water, it will again contain excess nitrates, and eventually that runoff water runs into our waterways, our creeks, our rivers, and eventually into coastal waters. And again, that can contribute to these harmful algal blooms. So what some farmers do is they run their runoff irrigation water or rainwater through what's called a bioreactor, and the bioreactor contains lots of soil microbes that can carry out nitrate reduction. So by running the water through the bioreactor, denitrification occurs. We're going to reduce the amount of nitrate in that water, and then after the runoff water has been treated in the bioreactor, it then will be released into the streams in coastal waters with reduced usable nitrates, and that again is going to reduce those harmful algal blooms. Another alternate terminal electron acceptor in anaerobic respiration would be sulfates. So sulfates can be reduced all the way to hydrogen sulfide. And this is going to happen in waterlogged soils like swamps, marshes. In sewers, we can smell it too. And the hydrogen sulfide gas smells like rotten eggs, right? So whenever you visit a swamp or a marsh and you smell that rotten egg smell, you know that's hydrogen sulfide being produced from anaerobic respiration. This third example, you guys, CO2 and carbonates, we're going to skip it just so we aren't totally overwhelming you. I mean, this is fascinating, however, because some of our domain archaea can use CO2 carbonates as terminal electron acceptors, and as a result, they'll make methane. So these guys are methanogens. They produce methane, and the reason that's important is that methane is one of our greenhouse gases that we think, as it's accumulating, is contributing to global climate change. But I won't have this on lecture exam too. One thing that's really cool is some bacteria are metabolically so versatile, and our kind of poster bacterium for metabolic versatility is good old E. coli. So for example, if E. coli is growing aerobically, it can carry out aerobic respiration, making the theoretical max of 38 ATP per glucose. However, if E. coli is growing anaerobically, for example, in our intestine, the intestinal lumen is anaerobic, it can switch over to anaerobic metabolism, and it has two choices. If there's an alternate terminal electron acceptor available, like nitrates, it can switch over to anaerobic respiration. And again, it won't make as much ATP as aerobic respiration, but using anaerobic respiration, it'll make more ATP than the 2 ATP per glucose that it would make during fermentation. If there's not an alternate terminal electron acceptor available for anaerobic respiration, then E. coli still has fermentation. So it can switch to fermentation and make 2 ATP per glucose, and it can survive just fine on that. Okay, folks, I think what we're going to do, let me close this, and let's see if I can't get this published on YouTube so that everybody can access it. Okay, and then we'll do another follow-up on photosynthesis and a final one on the nitrogen cycle.